Hi, everybody. As your AP Biology teacher, Mr. Poser. Today, we're getting into AP Biology topic 4.3, and that is on signal transduction pathways. So we got our introduction to signal transduction pathways in our last topic. We talked about the components of a signal transduction pathway. We talked about what transduction is, how it's basically how a cell internalizes, brings in a signal that it receives in order to cause a cellular response. And today, we're going to be getting into a few examples of different alterations to signal transduction pathways and how that can change the response to a signal. And we're going to talk about a few different responses to signals that cells can receive as well. All right, and we'll get into detail about a few different kind of examples of this happening. And we'll get into the specifics of one particular pathway. All right, it's not necessary to memorize this pathway, but it's nice to illustrate how transduction pathways work, and it covers a lot of the topics that we talked about in our last video here. Okay, so once again, here's an overview of cellular communication. We got our ligand over here. It binds to a receptor on the surface of a cell. Sometimes receptors can be inside of the cell, but this one's on the outside. All right, and then the conformational change, basically the change of shape of the receptor as a response to the binding of that ligand, triggers a whole bunch of different stuff to happen. That could be the release of second messengers, activating different kinases, and kinases activating different enzymes, and etc. It can get pretty complicated. Hey, but we talked about the general what happens with signal transduction pathways, the types of molecules that you're going to find in this kind of domino effect or this relay race type thing that you're going to see. We talked about it in the last video. All right, and then eventually here, the cell is going to respond to that signal as a result of that being brought in, right? So this is just complicated arrays of chemical reactions, right? A cell can't think, right? It doesn't receive a signal and say, I better make more of this protein. No, it's a chain of chemical reactions. This is a chain reaction where one enzyme is going to be particularly activated that will result in, a cell responding. That's the desired response to that signal. So here's the thing about signals. They can produce a whole lot of different responses, and some of those signals end up producing cellular responses that can change everything about what a cell does. So as I put here, a signal that a cell receives and transduces can change the cell's function and its gene expression. And this is a big point, okay? We're going to talk about a lot of different examples where a cell receives a signal and then transduces that signal, brings it inside, and what that results in is an alteration in the way a cell uses its DNA to make proteins. All right, it chooses basically which genes it's going to express and which genes it's not going to express. In other words, like it's going to choose which proteins it's going to produce and which proteins it's not going to produce based on what its job is. Okay, so if you think about this, I don't want to get too much into this topic right now because I'm sure it's going to come back later in the curriculum. Okay, but every human being, we start off as one cell, right? Where a sperm and egg fuse, and then you become one cell, right? And then that cell divides, right? And then you end up with like 3 trillion cells, right? But are you just a bundle of 3 trillion cells? No. You're a complicated system of tissues, organ systems that are all working together and doing their one job in order to maintain homeostasis and keep you alive, right? So brain cells are very different from intestinal lining cells, which are very different from liver cells, which are very different from cardiac muscle cells, which are very different from bone cells, right? And why are those cells different from each other? It's because they basically, I'm going to put this in air quotes, they pick and choose which proteins they're going to make and which proteins they're not going to make. Okay, so for example, a brain cell is not going to produce, proteins that are associated with muscle contraction, right? Bone cells are not going to produce digestive enzymes or anything like that. Okay, so different cells make different proteins. All right, and here's the why, right? Their gene expression, which genes they're going to actually use and which genes they're not going to use, are directed by cellular signals. Okay, so as I put down here, during development, cells differentiate, meaning they get a job and they get really, really good at just doing one thing. They become specialized at one function based on gene expression, which proteins they make and which proteins they don't. And what proteins they don't and do make is directed by signals. All right, a stem cell, as it's called, will receive a signal and say, hey, you're going to start making these proteins, all right, and then you will become a bone cell. You will become a blood cell based on the fact that you're making these proteins, all right? This blood cell is going to be making hemoglobin, and this bone cell is going to be making, fibrinogen, that kind of stuff, or not fibrinogen. Collagen. There we go. So that's something that's going to come up later on. All right. Another response that cells can take to a signal is what's called apoptosis. Okay, which is programmed cell death. Now, we're going to talk about a couple circumstances under which this is necessary later on, right? But sometimes cells, when they get a signal, they're basically told to burst, to blow themselves up, to self-destruct. Why is this necessary? Well, I mean, if this cell is super damaged, or it's cancerous, or it's going to cause some other issue and it's, beyond repair, sometimes a cell will receive a signal that will cause a chain reaction, that will cause signal transduction, that will result in the lysosomes basically digesting everything and going like that. And that's called apoptosis, all right? That is, that's the red button in certain circumstances that we're going to be talking about later on. Another point I want to make here is that alterations to signal transduction can result in different cellular responses, all right? So we talked about apoptosis being a cellular response. We talked about gene expression, alterations to gene expression being a cellular response to a signal, okay? But a cellular response can be completely changed based on if the particular protein is mutated or if a chemical is in the way that can interfere with the signal transduction pathway and the relay of a signal from when the receptor to the desired cellular response. If something gets bunked up in the middle, all right, that cellular response could be completely changed. It could not happen at all, or it could have a completely different cellular response that was not intended in the first place. All right, and mutations, which are alterations to the shape of a protein based on genes, or particular chemicals like tetrodotoxin over here, or a compound produced by anthrax, can cause alterations in proteins. Will be them receptors, kinases, enzymes, whatever, and that could result in changes in signal transduction and not producing the right cellular response or something completely different. Okay, so shout out to Mr. Anderson here for these two examples, right? There's particular toxins that are going to could interfere with voltage-gated ion channels, and those voltage-gated ion channels could result in neuron function not happening properly. So tetrodotoxin, which is released by pufferfish and particular types of newts, it's super toxic because it'll shut down your nerves basically, and yeah, that's how it'll kill you. And then here's another type of bacteria over here. It's called anthrax. All right, the anthrax produces a compound that interferes with adenylate cyclase. All right, and adenylate cyclase, if you recall from the last topic video, it makes CAMP, right? Cyclic AMP, our second messenger, or one of our main second messengers. And therefore, signal transduction can't really happen there either. So there you go. There's two examples of how chemicals can alter signal transduction pathways and result in different cellular responses. Okay, so just to illustrate this here once again, all right, this is a generalized signal transduction pathway. You got a whole bunch of stuff here. I don't know exactly what it is. I'm not going to lie to you. Okay, but all I'm trying to illustrate here is that if you let's just say you change the shape of the receptor, the ligand's not going to be able to bind and nothing's going to happen downstream. Or if you even mess with a kinase here, this message is not going to get relayed to result in the appropriate cellular response, etc. That's one of the main points I'm trying to make here. All right, so what we're going to end this video with here, I'm going to talk about a few examples of how gene expression can be affected by alterations to signals, or excuse me, they can be affected by what signals they receive, or alterations to signals can result in different gene expression and different cellular responses. All right, one of which we already talked about, quorum sensing. All right, if you recall what quorum sensing is, it's basically a way for bacteria to regulate how often they divide, right? Bacteria are single-celled organisms. They live in a colony, but you can't have too many of them in the same place because then they're not going to have enough resources and they're all, they're going to run out and they're going to die, right? So quorum sensing, as I put down here, is an example of how chemical messages can affect gene expression. The more densely populated a bacterial colony becomes, the more chemical messages are released that directly affect the expression of genes linked to binary fission, which is the cell division of bacteria, right? So over here, we don't have that many bacteria, right? And they're releasing this, every time they divide, they release this blue chemical over here. I don't know what the name of it is. Maybe you can look it up, but they release this blue chemical over here, all right? And then as more bacteria divide, it produces more of this blue chemical, all right? And the concentration of this blue chemical is directly affected, or excuse me, it directly affects the expression of genes here, all right? These are circular pieces of bacterial DNA that we'll talk about later on. They're called plasmids, all right? But you can see these blue molecules over here latching themselves onto the DNA and affecting how those bacterial cells are going to produce proteins, especially the ones that allow it to, well, make a copy of itself, divide, okay? So these blue molecules here, if they build up in too high of amounts, they're going to basically inhibit the expression of protein, or the expression of genes, the production of proteins that allow those bacteria to keep dividing. Okay, another example of how signals result in a cellular response, and depending on what that signal is, it can result in different cellular responses, is adrenaline or epinephrine. You might know adrenaline from the fight or flight response, right? Adrenaline is like when something really intense is happening, all right? Your fight or flight response is when your body is preparing itself for action, right? Whether that's to fight for your life or run for your life, right? So early humans, we use this, or animals in general, what they do, right? If there's some kind of something that could be perceived to be life-threatening, right? So you're walking through the woods and you get this bear all of a sudden, right? You know, your body is going to prepare you to either fight for your life or you're going to run for your life. Okay, you're going to square up or you're gonna get out of there. Either way, you need a lot of energy for that. You need a lot of awareness. Your heart rate's going to increase. Your breathing rate's going to increase. Your digestion's going to slow down, right? And it's going to prepare you for action. Okay, and epinephrine is a hormone, is a ligand that's going to bind to particular cells and cause a cellular response that, you know, gets your body ready for that. All right, and we're going to look at one pathway here today. Again, this is not necessary to really memorize, right? But it illustrates a lot of the points from the last topic, so we're going to get into it a little bit here. I've seen some practice questions on practice exams, maybe topic questions that have to do with epinephrine. So it's good to be familiar with it, right? But I would not say it's necessary to memorize all this as of right now. Okay, but here's what happens, right? Epinephrine is a ligand, all right? Let's just say this is the outside of the cell, this is the inside of the cell. We have a GPCR over here. This is the epinephrine receptor. I didn't label that. And down here we have some glycogen. And if you remember from Unit 1, glycogen is stored carbohydrates, right? In animals, right? So we have a polysaccharide over here, and each of these hexagons over here is a glucose, and they're bound together, right? They have not peptide bonds, covalent bonds, linking those individual molecules together, all right? And if we're getting ready to fight for our lives, we're going to need a lot of glucose so that we can make ATP. Our mitochondria can make ATP, bind to this receptor over here. All right, so here's epinephrine. It's going to go on to trigger is the phosphorylation of this alpha subunit of this protein down here, right? Remember, so G protein coupled receptors, that's what GPCR stands for. The G protein is this set of 3 circles right here. There's alpha, beta, and gamma. The only one that we need to worry about right now is the alpha subunit of this protein. Hey, when epinephrine binds to the receptor, it basically causes the change the shape of the receptor, the change of the shape of the protein, and triggers GTP, which is related to ATP, to phosphorylate this alpha subunit over here. All right, so check it out. It has a whole bunch of energy. It is activated. It is phosphorylated. And now it can do some stuff. Okay, and what is it exactly going to do? Well, it's going to float on over here and activate adenylate cyclase. Does anybody remember what adenylate cyclase does? Well, it takes some ATP and produces our friend cyclic AMP. Our cyclic AMP is our our second messenger, right? And it can activate a whole bunch of other different pathways, right? So imagine a whole lot of cyclic AMP being made so that it can activate a whole lot of different pathways at the same time, right? Epinephrine doesn't just cause one response, okay? But this is the one response that we're going to be looking at here for today. All right, so once CAMP is produced, it's going to activate this kinase over here. And if you recall from the last video, kinases are proteins that just basically pass a phosphate along and activate something else. So CAMP is going to activate this kinase, and then this kinase is going to move over here and activate glycogen phosphorylase. All right, glycogen phosphorylase. All right, it receives a phosphate group. And guess what it's going to do? Well, it's going to hydrolyze glycogen. So here it goes. It's going to go over here, bing, bing, bop, and hydrolyze those covalent bonds. And it's going to turn that glycogen into 4 different glucose molecules. All right. Now, glucose gets the body ready for producing a whole lot of ATP really fast. All right. And that's part of the fight or flight response, is that you got to have a whole lot of ATP. Now, there's a whole lot of different things that happen as a result from epinephrine, right? And that's largely linked to, CAMP activating a whole lot of different pathways. But we're just focused on one that's going to get your body cells ready for producing a whole lot of ATP. All right, and basically accessing that stored glycogen and breaking it down to get a whole bunch of glucose. Okay, a couple other examples of signals altering gene expression. Cytokines. This is a result of the immune system, right? So let's just say we got our good old friend SARS-CoV-2 over here, which is a virus, okay? And let's just say it enters one's body here. What it's going to trigger, those proteins here on the surface of that SARS-CoV-2 capsid, what they're going to trigger here are inflammatory responses, all right, in a whole bunch of these other cells. And they are going to release a group of signaling molecules called cytokines, okay? And cytokines can get really, really super complicated. Again, immune system is like the most complicated thing ever besides the human brain. But basically what's going to happen here is that cytokines are signaling molecules that are going to be released in very large amounts in order to activate in what's called an inflammatory response. It's going to activate other cells to prepare to get rid of that SARS-CoV-2 that has entered your body. Okay, your immune system works really, really hard to protect you. Shout out your immune system. But as I put down here, cytokines are signaling molecules that are used by immune cells when an infection is detected. This triggers other cells to begin an inflammatory response. All right, so basically when those other cells receive a cytokine signal, it's going to start expressing genes. It's going to start making proteins that will allow it to respond to that SARS-CoV-2 and hopefully fight that off. Okay, so here we go. And here's another example of how signaling molecules can affect gene expression. Yeast cells. So yeast are single-celled organisms, right? They're classified as a fungus, right? But they're single-celled organisms. And they can reproduce sexually, which means that they can exchange genes with another cell, all right, and produce some genetic variation, right? It takes 2 to tango, right? I know that's weird to think about when it comes to yeast cells, but here you go, right? Pheromones, all right, which are another group of signaling molecules that are going to tell other cells, hey, I'm here. They're going to communicate. So you might have heard of this with like dogs or like body odor stuff before. Usually pheromones are associated with mating, right? So they're going to be signaling molecules that can communicate with other cells and basically prepare them to mate. In other words, so you might have heard like dogs, like, marking trees with pheromones or whatever, or like certain animals are in heat or whatever. They're going to be releasing pheromones that other animals are going to pick up, and that communicates information. So pheromones are also signaling molecules. Okay. In yeast cells, what it happens to do, one yeast cell releases pheromones, which are going to get picked up by receptors on another yeast cell, and it's going to cause the expression of genes that allow it to get ready for sexual reproduction. And I've learned a new word. One of my favorite parts about this job being a teacher is that I also get to learn new things. So as I was researching pheromones in yeast sexual reproduction, I found one of my favorite words ever. It's called a shmoo. Here it is. The shmoo formation is the direct result of pheromone signaling in yeast cells, in a particular type of yeast cells. And the shmoo eventually becomes a bridge between these two cells that allows them to exchange their genetic information. There it is, a shmoo. So take that back to your biology class. If you're watching this at home, take it back to class tomorrow that you learned what a shmoo is from Mr. Poser's video here too, because that's the best word ever. So there you go. Okay, another example of how signals can result in differential gene expression. Okay, and then the last thing that we're going to talk about here is that when fruit ripen, they release a compound called ethylene, and ethylene results in the expression of proteins on neighboring fruits. That allows them to ripen. All right, and ripening is the breaking down of particular components that like cellulose in plants, for example. It's going to start to break that down, some proteins. And it makes it easier for us to eat. It makes it easier for us to digest it when fruit is ripened. And ethylene is well, you don't want it to ripen too much, right? Because then it like rots and stuff, right? But when ethylene is released, it causes plants to ripen, okay, or fruits to ripen. And that is basically a response to a cellular signal with differential gene expression. Okay, so we're going to talk. This is also an example of positive feedback. We're going to talk about that pretty soon, right? So like one banana, if it starts to ripen, it's going to release ethylene, which will cause another banana to start to ripen and release ethylene, which will cause another banana to ripen and release ethylene. And that's part of the reason why bananas ripen so fast. But anyway, then we'll talk about that later on. Here's the recap. All right, there's only a few main points. We talked about a whole lot of examples, but here it is. Responses to signal transduction can vary. It can result in alterations to gene expression, cell function, or it can even result in apoptosis, which is programmed cell death, right? But that gene expression part is very, very important. But depending on what kind of signal a cell receives, it might change what genes it expresses. In other words, what proteins it makes, what proteins it doesn't. Sometimes it can just get told to blow up, and we're going to talk about that later on. Changes in signal transduction, like mutations to receptors and kinases or chemicals, can alter, inhibit, or activate pathways. So there you have it. Okay, if you mess with a particular part of a pathway, you can either shut that pathway down, activate another pathway, or get something completely different. And we talked about a whole lot of different examples of signaling and responses, especially those with gene expression. Okay. That is it for this video. Please let me know if you have any questions. We'll see you next time.